Hello and welcome everyone to this podcast for genetics. In the past several podcasts, we focused on gene regulation in bacteria. This will be our first of a few podcasts where we talk about eukaryotic gene regulation. Now, there are several similarities when we think about eukaryotic and bacterial gene regulation, but the biggest one is that both bacteria and eukaryotes use DNA-binding proteins to regulate gene expression. They regulate their genes in other ways too, but remember, bacteria primarily use these DNA-binding proteins to regulate which genes get transcribed and which genes don't. Eukaryotes will also use this mechanism, but as we'll see, they use many other mechanisms as well. Now I want to jump into some of the differences between eukaryotic and bacterial gene regulation, which will also set up the different things we're going to talk about over the next few podcasts. So, differences between eukaryotic and bacterial gene regulation. Okay, the first thing I want to talk about is gene organization. And what I mean by this is that in prokaryotes, remember, you would have one promoter that regulated several genes, and it made an RNA molecule that had each of these genes. We'll call them A, B, and C. I didn't mention it at the time, but what we call this is polycistronic messages. Poly, because we have more than one gene represented on this single RNA message. Counter to that, usually in eukaryotes, there will be a single promoter that regulates a single gene. Maybe it's gene A. It will make a single RNA molecule that only has the coding information for gene A. And so we call this a monocistronic message, because there is one gene represented in this one RNA message. A second difference is that eukaryotes have a complex chromatin structure. If you remember, earlier we said the term chromatin is a term that we use mainly to talk about eukaryotes. And that's because it can exist as a euchromatin or heterochromatin. Those terms don't exist in bacterial chromosome discussions. Because of this complex chromatin, histones are added or removed to regulate genes. A third reason is the presence of a nucleus in eukaryotes. Remember, you have your eukaryotic cell here with its nucleus. All the DNA is in the nucleus. The messenger RNA is going to be made in here. And so we have to get that messenger RNA out of the nucleus and into the cytoplasm in order to make a protein. So that adds a level of complexity that we see in eukaryotes that we don't see in bacteria. The next reason is that many eukaryotes, not all, but many eukaryotes are multicellular. So let's just look at a brief example here. Let's think about a human, and in humans we have many different cells and many different types of tissues. And so let's look at a cell that's in the pancreas. And then let's look at a cell in the eye. Each one of these cells will have the exact same number of genes in them, but in the pancreas we want to express, and I'll just show that by an up arrow, the gene for insulin, because the pancreas makes insulin. In the eye, we want to express genes such as crystalline. Not anywhere in the eye, but in the lens part of the eye. These are transparent proteins. Now, in the pancreas, we don't want to express crystalline, so I'm going to put a down arrow for down regulation. And in the eye, we don't want to express insulin. So because of this multicellularity, there's an extra degree of regulation necessary so that the pancreas behaves as a pancreas and the eye behaves as an eye. In general, we can think of eukaryotes as having a more complex development. So a bacterial cell has to regulate its genes to survive, and it has to do so in a very rapid way. But from the time it forms till the time it dies, the overall structure and complexity of the bacterial cell doesn't change much. However, in eukaryotes, there's a very elaborate developmental pathway. So let's use humans as an example. First of all, we make gametes, eggs and sperm, And these eggs and sperm form that first embryonic cell, the zygote. And then, through an elaborate process during embryogenesis and through fetal development, you will make a fully formed human being. And obviously, I left out a lot along this pathway. If you want to learn more, you should take a developmental biology course where you can learn about the fascinating things that happen from this single-cell zygote to this fully formed organism. And then even after we're fully formed, we go through an aging process that will ultimately lead to a programmed cell death for our cells and ultimately for the organism itself. So we have a much more complicated development. The last difference I want to talk about is the source of genes. This is an emerging field, and we'll talk a little bit about this as we move forward. But which genes get expressed or not expressed is often determined by whether or not that gene is inherited from the dad or from the mom. So I'm going to erase this, and then I want to draw a eukaryotic cell. And in that cell, I'm going to talk about the different levels of gene expression. And this large cell here will serve as our roadmap for the next several podcasts. So here's our large cell. And we can make it our pancreatic cell that we've talked about in the past, and we'll probably continue to talk about several times. And let's draw a rather large nucleus here. And in this nucleus, as no surprise to you, we'll have all of the DNA. And within this DNA are several genes. We'll just draw 2 for now. And just to make it more real, let's just say this is the insulin gene, and we'll make this the crystalline gene. Now in reality, these 2 genes aren't right next to each other, but for simplicity, I'm just going to draw it that way. Now if this is a pancreatic cell, we only want to turn on the insulin gene and not the crystalline gene. And so regulation at the level of transcription is very important. And within this level of transcription, there are several things we should consider. How the histones are added and removed is one consideration. Modifying the DNA is another mechanism to regulate genes at the level of transcription. And that's often caused by methylation. Regulating whether or not a transcription factor can bind. And a lot of what I'm talking about here is in the field of epigenetics. These are all important considerations when we think about how this gene is going to be regulated at the level of transcription. Now, as this gene is transcribed, it will make a messenger RNA, 5' to 3'. And that messenger RNA, as we've learned about, is processed. The introns are removed, the poly A tail is added, and the G cap is added. And this is the second level that genes can be regulated. We'll talk a little bit about this in a future podcast. I should have mentioned we'll talk in great detail about these things in future podcasts as well. The next level of regulation involves nuclear export. Some genes can be regulated in such a way that the messenger RNA is transcribed, it's processed, but it hangs out in the nucleus until it's needed to be moved to the cytoplasm to make proteins. So this is our third level of gene regulation. So this messenger RNA will be in the cytoplasm, and it will have 2 fates. It can be translated using the ribosome and the other factors we talked about. And whether or not it gets translated or not is a fourth way that genes can be regulated. Now, at some point, this messenger RNA can be degraded. This can be an active process where this RNA molecule is targeted by other RNA molecules to be degraded, or sometimes it's just the loss of the poly A tail that makes it unstable. And we'll spend a little bit of time talking about this as well towards the end of this series of podcasts on eukaryotic gene regulation. So this messenger RNA is translated, and it makes this protein. Sometimes this protein is active and ready to go, but often it is inactive. Now, and we've mentioned this before, but sometimes this inactive protein will then undergo a post-translational modification to make an active protein. Sometimes this change is adding chemicals to it, like phosphate groups or acetyl groups or other chemicals. Or sometimes it Just helps it fold a little bit better. And so this is the sixth way genes can be regulated. And the final way that I'm going to talk about in this slide here is through protein degradation. Proteins have a certain half-life. They don't survive forever. At some point they are degraded. That is a way that we can effectively shut down the expression of this gene. So that's our seventh way. As I've hinted along the way, we're going to spend a great deal of time talking about these mechanisms. We'll talk a little bit about the processing, and we'll talk a little bit about the degradation of the RNAs as a way to regulate the gene expression. The rest of these you should know, but just at the level that I've described right here on this board. Okay, so that's all I'm going to say on this podcast. This podcast was just to introduce you to eukaryotic gene regulation. On the next podcast, we're going to focus in on these histones and transcription factors. If you have any questions, let me know. If not, I'll see you in class.